Point mutations. Point mutations involve the substitution of one nucleotide base for another. There are 4 types: missense, nonsense, silent, and read-through. These point mutations are also categorized as transitions or transversions. Transitions include purine to purine base changes and pyrimidine to pyrimidine base changes, while transversions include purine to pyrimidine base changes and pyrimidine to purine base changes. To review, purines are nucleotide bases adenine and guanine, and pyrimidines are nucleotide bases cytosine, thymine, and uracil. Examples would be purine to purine adenine to guanine, pyrimidine to pyrimidine cytosine to uracil, and pyrimidine to purine cytosine to adenine. The following DNA strand is translated into an mRNA strand. TGA on the DNA strand is read as UGA, which is a stop codon, so translation does not continue past this codon, leaving the rest of the codons on the non-mutated mRNA strand code for methionine, tryptophan, asparagine, alanine, and cysteine. Now, a missense point mutation takes place, changing this cytosine in the DNA strand to an adenine. This is an example of transversion. Starting with the 5' end of our new mutant mRNA strand reads AUG, UGG, but then the next codon is now AAA instead of AAC. This new AAA codon now codes for lysine instead of asparagine, which can either alter the protein's function, make it non-functional, or have no effect. The next type of point mutation we will look at is nonsense, which changes this cytosine into an adenine, which is once again a transversion. Instead of UGC coding for cytosine, the strand now reads UGA, which is a stop codon. Nonsense mutations like this that cause premature stop codons can have varying effects on the protein. However, silent point mutations have no effect on the protein because they code for the same amino acid. For example, if this thymine was changed to a cytosine, which is an example of a transition, there would be no change to the amino acid sequence. The amino acid sequence would be the same because GCC and GCU both code for alanine. Finally, the last type of point mutation is a read-through, which means that the stop codon is changed to code for an amino acid. For example, when I change this thymine to an adenine, which is a transversion, the sequence continues instead of stopping. The old stop codon, UGA, now reads AGA, which codes for arginine. This change will completely change the amino acid sequence, resulting in a longer protein.